ELISA is the basic assay technique, known as enzyme-linked immunosorbent assay, that is carried out to detect and measure antibodies, hormones, peptides and proteins in the blood. Antibodies are blood proteins produced in response to a specific antigen. It helps to examine the presence of antibodies in the body, in case of certain infectious diseases. ELISA is a distinguished analysis compared to other antibody assays, as it yields quantitative results and separation of nonspecific and specific interactions that take place through serial binding to solid surfaces, which is normally a polystyrene multiwell plate. ELISA tests can be classified into three types depending upon the different methods used for binding between antigen and antibodies, namely, indirect ELISA, antigen is coated to the microtiter well; sandwich ELISA, antibody is coated on the microtiter well; and competitive ELISA, microtiter well which is antigen coated is filled with the antigen-antibody mixture. Indirect ELISA detects the presence of an antibody in a sample. The antigen is attached to the wells of the microtiter plate. A sample containing the antibodies is added to the antigen-coated wells for binding with the antigen. The free primary antibodies are washed away and the antigen-antibody complex is detected by adding a secondary antibody conjugated with an enzyme that can bind with the primary antibody. All the free secondary antibodies are washed away. A specific substrate is added which gives a colored product. The absorbance of the colored product is measured by spectrophotometry. Sandwich ELISA helps to detect the presence of antigen in a sample. The microtiter well is coated by the antibody. The sample containing the antigen is added to the well and washed to remove free antigens. Then an enzyme-linked secondary antibody, which binds to another epitope on the antigen, is added. The well is washed to remove any free secondary antibodies. The enzyme-specific substrate is added to the plate to form a colored product, which can be measured. Competitive ELISA helps to detect antigen concentration in a sample. The microtiter wells are coated with the antigen. Antibodies are incubated in a solution having the antigen. The solution of the antigen-antibody complex is added to the microtiter wells. The well is then washed to remove any unbound antibodies. More the concentration of antigen in the sample, lesser the free antibodies available to interact with the antigen, which is coated in the well. The enzyme-linked secondary antibody is added to detect the number of primary antibodies present in the well. The concentration is then determined by spectrophotometry. ELISA is one of the easiest blood tests that can be carried out. It is rapid, quick and requires a blood sample of the patient. The entire procedure of ELISA is mentioned below. An antibody is attached to a polystyrene plate which is a solid surface and is attracted or has an affinity towards bacteria, other antibodies and hormones. A microtiter coated with antigen is filled with this antigen-antibody mixture, after which free antibodies are removed by washing. A second antibody specific to primary antibody is added, which is usually conjugated with an enzyme. Free enzyme-linked secondary antibodies are removed by washing the plate. Finally, the substrate is added. The substrate is converted by the enzyme to form a colored product, which can be measured by spectrophotometry. HCG protein, which indicates pregnancy, is detected by ELISA. A combination of blood or urine sample and purified HCG linked to an enzyme is added to the system. If HCG is absent in the test sample, then only the linked enzyme binds to the solid surface. The more the substance of interest is present, the more reaction takes place and less of linked enzyme binds to the solid surface. These reactions are indicated usually with a change in the color of the solution.